Good evening, everybody. Good evening, Gyanendra. Okay. So today is week 8, and we will start our live session with the topics. And this topic are cell biology and microscopy. So basically, we will try to understand what do you mean by cell, what different component, what different organelles we have in the cell, what are the different functions a cell can do, then how we can see the structure, the organelles of the cell using some instrument, for example, using microscope, advanced microscope from like electronic transmission. So we will try to understand the basic structure and function of the cell, how this cell structure is different in prokaryote, in eukaryote, in archaea, and how we can see such kind of structure using some instruments. So we have a lot of question based on this cell biology and microscopy. So let me start with first. So the first question is, which of the following correctly describe the stabilization of protein tertiary structure? So let me please tell you that protein have four kind of structure: primary, secondary, tertiary, and quaternary structure. So, and this question is based on the tertiary structure. So you have to tell me how this tertiary structure gets stabilized. So I have four options. Tertiary structure is stabilized only by hydrogen bond. B is tertiary structure is formed by interaction between multiple polypeptide chain. C is tertiary structure is stabilized by ionic bond, covalent bond, hydrogen bond, and hydrophobic interaction. And D is tertiary structure refers to the sequence of amino acid in a polypeptide chain. So you have to select the correct one. You people can write in the chat box, and as well as you can communicate while uh, removing this mute, and then you can speak the answer as well. Okay, so answer will be C, and let me explain you why. First, you have to get in mind what do you mean by this protein, what is the structure of the protein. So let me show you this. So the basic component of the protein is having three things: amino group, side chain, and the carboxylic group. So let me show you. So this is a single component of the protein. When this component will combine in a sequence, that will be known as protein. So this is something amino acid. So this structure is for the amino acid. Amino acid. So in amino acid, we have amino group, that is NH2, then one carbon. This carbon will have R, that R can be anything. We will tell about this. And then we have the hydrogen and one carboxylic group. So if we have such kind of structure, then we will call it as a amino acid. Okay, when we have multiple amino acid attached to each other in a linear way, or you can say using the polypeptide bond, then this will be the structure of protein. Okay, so you have amino acid 1, you have amino acid 2, 3, 4, 5, and if we are bonding to them together in a sequence, then it will be a protein. Okay, and this R may be anything. So if R is H, if this R is H, then this amino acid will be known as glycine. If this R is CH3, then this amino acid will be known as the alanine. So likewise, if we are changing the component, this R, CH, CH3, CH3, and if we are changing this, if we are advancing this with some other structure, replacing this R with some other carbon and hydrogen bond, then this will give you the different kind of amino acid. And if you will see, we have total 20 amino acid. So all the protein, whatever we have in our body, is composed of 20 amino acid, right? Okay, so just keep in mind right now, we have amino acid. If this amino acid or amino acid are attached to each other using a polypeptide bond, then this will make the protein. Okay, so this is the one thing. Then, okay, so if these amino acid are in a single chain, if these amino acids are in a single chain, then this will be the primary structure of the protein. So this will be the primary structure. So glycine, then valine, glutamine. So if this amino acid are in a single structure, then this will give you the primary structure of the protein. So this is primary structure of the protein. This is what primary structure of the protein. Okay, then in that primary structure, if amino acid and amino acid will connect each other, this can be by sulfur bond as well. But just keep in mind, if we have the single chain, then this will form the primary structure of the protein. Okay, then let me show you. Then this is the secondary structures. What will happen? So these are the chain of the amino acid. So this is one chain of the amino acid. This is another chain of the amino acid. And if we do have this hydrogen bond, if we do have this hydrogen bond attached with two lines, then this will give you the secondary protein structure, and this can be a beta plated sheet, and this can be alpha helix as well. So what you got is, you have amino acid. If this amino acid will be attached in a linear way, then this will be primary structure. If this will be connected to each other using this hydrogen bond, then this will give you the secondary protein structure. Okay, but the component is amino acid or amino acid, right? Then what will happen? This is the tertiary structure what we were talking about. So this is the chain of amino acid. So this is the chain of amino acid, and where they can be connected by the ionic bond. They can also connect by the polypeptide. This is backbone, and this will give you the primary structure. But whenever you have the ionic bond, whenever you have the disulfide linkage, whenever you have hydrogen bond, whenever you have hydrophobic interaction, if such kind of bonds we have in that chain of the amino acid, then this will be known as the tertiary structure of the protein. Okay, and the question was like that only. So let me show you the question. So question was stabilization of the protein tertiary structure. So this is stabilized by the ionic bond, covalent bond, and hydrogen bond, and the hydrophobic interaction. So you learned that we have amino acid. The chain of amino acid will give you the primary structure. If we have the hydrogen bond between that chain, then this will give you the secondary structure. If we do have the ionic bond, covalent bond, hydrogen bond, hydrophobic interaction within this amino groups, then this will give you the tertiary structure. And we also have one quaternary structure. This is like whenever we do have different polypeptide chain, then this will give you the quaternary structure. So we have a primary, secondary, tertiary, and quaternary structure. So and let me tell you, the hemoglobin is nothing but a protein, and that protein have the quaternary structure. Okay, and because of that quaternary structure, that protein is very specific in nature. So just keep in mind, if we have the hemoglobin, this hemoglobin is nothing but the protein, and this protein having the quaternary structure. Okay, so let me move to the next question. So next question is, which of the following statement about biomolecular chirality is correct? And options are L sugar and D amino acid are most commonly found in nature. B is most naturally occurring sugar exists in the D form, while most naturally occurring amino acid exists in the L form. C is D amino acid are the building blocks of the protein in living organism, and D is sugar and amino acid do not exhibit chirality. So, what do you think? What is the right answer for this? So, if you people know that we have one structure, so let's say this is the one structure, and this structure may have the mirror image. So, this will be the mirror image, not like this, but this. So, this is the mirror image. The question is like, so we have amino acid and sugar. So, which form, this one or this form, is most common? So, you have to tell me about like this. Uh, Okay, let me show you. The answer will be B, because like whenever we are talking about the sugar, then sugar exists in the D form. I will tell you, I will explain you what you mean by this D form, and amino acid will exist in L form. So let me show you what you mean by this D form and L form. So just see here, this is the structure of glucose. This is the structure of glucose. Okay, so and if we have this OH, this one, so we have to see this OH. If this OH is in the right hand side, then this will be known as D glucose. If this is in left hand side, then this will be known as L glucose. Okay, so this is nothing but the kind of mirror image. So if OH in the right side, then this will be D glucose. If this is on left side, then this will be L glucose. Likewise, this is the amino acid. We talked about amino acid, right? So we have one C, and this C is attached to the carboxylic group. This C is attached with the amino group, and this is R, and this is H. So this is one amino acid, right? So this amino acid may also have the mirror image, and this, the mirror image of this one is this one. So H is near to this, and this one. So this is something like mirror image. So we have D form, we have L form. If we have that functional group in the right hand side, then this will be D form, and if this is on left hand side, so here you just see OH. If this is on left hand side, then this will be known as the L glucose. And my question was, which one is more common? So which one is more common? So whenever we are talking about sugar, then most naturally sugar will exist in the D form, and the amino acid will exist in the L form. So this is just you need to know. So based on that, we had that question. And you can also see here, this is again, and whenever we do have the mirror image, then this is known as the enantiomers, right? So this is how you can solve this question. Okay, next question is about the cell wall. So which of the following correctly describe the modification of cell wall precursor in bacteria? So we have two compound. The one is NAG and the second is NAM. So this is known as NAM and NAG. So you have to tell me like from where or from what compound this has been derived. So the options are NAG and NAM are derived from modified amino acids. NAG and NAMs are sugar modified by addition of acetyl and ether groups. NAG and NAM are modified sugar derivatives where hydroxyl group are substituted by acetyl and ether linked lactyl groups. And bacterial cell wall are made only of unmodified glucose molecules. So this is about in the cell wall of the bacteria, we have two component, or you can say we do have two chemicals that are NAG and NAM along with other components. So you have to tell me from what kind of compound they are made up of. So out of this four option, you can tell me one, and you can write in the chat box. I will see whether you are giving me right answer. And even if you are not giving me right answer, then it's okay. I will try to correct you. From the name of this NAG and NAM, N-acetyl glucosamine and N-acetyl muramic acid. So from that name as well, you can predict something about the original compound from where this NAG and NAM were derived. So, actually, if you will see here, glucosamine. So, what do you mean by this? Initially, it was the glucose, and glucose was modified. And how they were modified? Where hydroxyl group, we had the hydroxyl group that were replaced with acetyl and ether linked lactyl group. So, let me show you first the NAG and NAM. So, this is the glucose, right? This is the structure of glucose, where we have 1, 2, 3, 4, 5, 6. So we have 6 carbon, and we have this O double bonded H, and then we have HOH linkages, and at last we have CH2OH. And if we can write in the circular form, then we can write in this way. So this is like different kind of projection we can do for this D-glucose. So just keep in mind, this is our glucose structure, right? And so this was our glucose structure. So just see here, we just changed this one. We changed this one. We changed this one and this one. So what we are doing, we are replacing the hydroxyl group with something else here, but the basic structure of the glucose is same. So, and this will give you NAG, and this will give you NAM. So here we are just changing some groups at some point. So that's why this is the derivative of glucose only. But whenever we are let me show you. So these are glucose derivatives where hydroxyl group, these OH groups, are replaced by acetyl and ether-linked lactyl group. So from here you can guess, in the left hand side we have the glucose structure, but the derivatives are NAG and NAM, and here some groups were replaced by something. So this is how NAG and NAM were derived, and these are the important part of cell wall of the bacteria. These are very important structure that will give you the cell wall structure of the bacteria. Anyway, so next question is, so right now we are just talking about the biochemistry, because like whenever we are talking about the cell wall, cell membrane, or mitochondria, or chloroplast, or nucleus, first we have to know the basic structure of each of these component. So if we are talking about the nucleus, then what kind of chemical or component it is made up of. Or if we are talking about the cell wall, then what is the basic component of which this cell wall is made up of, because we have a lot of advantage knowing the biochemistry. For example, if we do know the chemical formula for this cell wall, then we can understand like what kind of receptor, what kind of antibiotic we may have that can attack that cell wall. So biochemistry is so important right now. So that's why we have a lot of question on firstly biochemistry, and based on the cell biology. So we have different component. So for example, we have mitochondria, chloroplast, and for each component, so what are the chemical that will make such kind of such kind of components or organelles. So the question here is, which of the following statement correctly differentiate between starch, cellulose, and glycogen? We already have talked about the cell wall. So in the last question only, we got to know that if we are talking about the cell wall of the bacteria, that is made up of NAG and NAM, right? But if we are talking about the cell wall of plants, then this is made up of the cellulose. If we are talking about the cell wall of the fungus, then this will be made up of the chitin. So we must know what is the difference between this NAG, NAM, cellulose, chitin, and even the difference between starch, cellulose, and glycogen. So we have four options here. Starch and glycogen have alpha, one for glycosidic bond, while cellulose have beta, one for glycosidic bond. B option is cellulose is highly biodegradable while starch is not. C is glycogen is a linear polymer of glucose while cellulose and starch are highly branched. And D is cellulose, starch, glycogen all serve as energy storage molecule in bacteria, plants, and animals. So here what we are doing, we are trying to differentiate between the structure of starch, cellulose, and glycogen. Not D, but something else. Okay, so the answer will be A. Let me show you how. So this is the basic structure of starch, glycogen, and cellulose. So if we do have the single chain, then this will be amylose. If there is like one branch, then this will give you the starch. If we have multiple branch, then this will give you the glycogen. And if we have like a layered and very parallel chains, then this will give you the cellulose. And if I show you the structure here, so just see here, this is the glucose. So either it is cellulose or starch or glycogen or chitin. These four are made up of the glucose. These are made up of glucose only. So see here, this is the glucose structure. This is the glucose structure. This is glucose structure. But how this one glucose is connecting with the another glucose will give you rise the difference between cellulose, starch, glycogen, and the chitin. So if you see here the bond, the important thing is to see here. So this one, how this is bonding here, this is different from this one, this is different from this one, and this is different from this one. So just see here, this glucose structure and this one. So this is here, this is in sequence. Here this is in sequence, but the bond, how they are bonding to each other will give you rise the cellulose, starch, and glycogen and chitin structure. So what we can see, let me clear it. So here just see the difference. CH2OH is here and CH2OH is here, and then they have this structure. Here water will be eliminated and this will give you the oxygen, right? And this is how this will be made up of. So this is the cellulose structure. Here if you will see CH2OH, CH2OH, so this is in a very uniform sequence, and that's how this will give you the starch. So likewise we have different structures, and but just keep in mind the unit for all these cellulose, starch, glycogen, chitin is glucose only, right? So if I'm showing you the, so here Starch and glycogen have alpha 1 for glycosidic bond, while cellulose have beta 1 for glycosidic. So this 1 and 4 is the place from where this bond were formed. So okay, the next question is about the significance of Reynolds number. So, in fluid dynamics, so you have to tell me what do you mean by this Reynolds number. So, I have four options. Reynolds number is the ratio of viscous force to inertial force and determine whether flow is laminar or turbulent. B option is a high Reynolds number indicates dominance of viscous force leading to smooth laminar flow. Reynolds number is given by the formula Nr is equal to inertial force upon viscous force, and D is low Reynolds number indicate turbulence while high Reynolds number indicate laminar flow. So you have to tell me the correct statement or correct significance of the Reynolds number. So the answer is C, and let me show you how. So this Reynolds number is actually dimensionless, so it doesn't have any unit, and this will tell you the flow behavior of the fluid by comparing the inertial and viscous force. So here, if you will see, this is the formula. So Reynolds number is equal to rho. So this is the density of the fluid, V velocity of fluid, D diameter of pipe upon viscosity of the fluid. So what is the significance of this? So for example, uh, we have a bacterial cell and we also have eukaryotic cell, and if we are talking about the movement of the of a particular cell, either it is of bacteria or the human cell, then we have to see whether it is like the movement is controlled by inertial force or by viscous force. Then you will get to know that for the bacterial cell, then the dominating force will be the viscous force, but for another the advanced cell, this will be the inertial force that will control the movement of that cell. So for that only this question was asked. Okay, then we have another question based on the bacterial and archaeal membrane lipid. So we have the question, that is, which of the following statement correctly differentiate between bacterial and archaeal membrane lipid? And the four options are bacterial and archaea both have membrane lipid with ester linkage. B is bacterial membrane contain ether-linked phospholipid, whereas archaeal membrane contain ester-linked lipid. C is bacterial predominantly have ester linkages in their membrane lipid, while archaea have ether linkages allowing them to survive extreme condition. And both archaea and both bacteria and archaea exclusively use the ether linkages in their membrane lipid. So you have to tell me how we will differentiate the cell wall of bacteria and archaea. So we can differentiate it using the membrane lipid. So you have to tell me what kind of membrane lipid we have in the bacteria and what kind of membrane lipid we have for the archaea bacteria. So the first thing is very clear. If we are talking about if we are talking about the cell wall or the cell membrane, basically here we are talking about the cell membrane, then cell membrane have the lipids either in the bacterial or in the archaeal cell membrane. Yeah, this is option is not B but C. Just keep in mind, if we are talking about the bacteria, then we have the ester linkages, and if we have the archaea, then we have the ether linkages. Okay, and because in the archy we have the ether, and this ether linkages is very important because that will allow them to survive extreme condition. If you will get to know the difference between archy and bacteria, then archy are more tolerant to the extreme condition, and this tolerance is given by these ether linkages. But if we do have the ester linkages, then this will differentiate this archaea from bacteria, normal prokaryotic bacteria. And let me show you how. Okay, so just see in the figure. So this we have the phospholipid. We have the phospholipid from bacteria and from archaea. This is the single chain. We have the branched chain here of the phospholipid. But the thing we have to see is this one. This is ester linkage and this is ether linkage. So for the archaea we have the ether linkage. For the bacteria we have the ester linkages. So that's how we can differentiate between the cell membrane of bacteria and archaea. If we have the ether, then this is archaea. If we have the ester linkages, then we will tell that this cell membrane is for the bacteria. Okay. Okay, the next question is, we have a cell, right? We have the cell membrane, and if something is needed to be entered, or if something is needed to be exit from that cell, then we must have some transport mechanism. And we do have two type of transport mechanism. The one is passive and another is active. And this question is based on passive transport mechanism. So you have to tell me the correct statement, and I do have four options. Simple diffusions allow larger molecule to pass freely through the lipid bilayer. So what we are talking, this is the cell membrane. So if this is the cell membrane, then how we can pass the molecule in and out? Okay, so the first is simple diffusion allow large molecule to pass freely through the lipid bilayer. The B option is facilitated diffusion require ATP to transport molecule across the membrane. C is aquaporins are specialized transmembrane protein that facilitate water transport, primarily function as water exporter. And D is osmosis is an active process requiring energy input to move water across membrane. So the question is just like that. If we do have the cell membrane, and if we need to import and export some nutrient, or import and export some water, then what kind of mechanism we have under this passive transport mechanism? And you have to tell me the right statement. So the answer is C. Let me, yes, Yogi, the answer is C. How this C is, uh, just very small molecule we can pass through simple diffusion. And if we are talking about diffusion, then this will not be the solid molecule. This must be gaseous in phase. So if we have the gas, if we just have the gas, then only this simple diffusion is possible. This is not possible for another kind of another phase of any molecule. If these molecules are in gaseous, or if it is in smaller molecule, then only this simple diffusion will work. Then if we are talking about the facilitated diffusion, then this is the passive transport and it doesn't require ATP. Then this is also the wrong statement. This is also the wrong statement. And osmosis is not the active, but this is the passive, and it doesn't require energy. So this is again the incorrect statement. The only correct statement is aquaporin. So you just have to see first the difference between passive transport and active transport. So whenever we need some energy for the transportation, so for example, if this is the cell and if we want to enter water into this cell, and if we need some energy for this transport, then this will be known as the active transport mechanism. And if we do not need energy to move this water from outside to inside of the cell, then this will come under passive transport mechanism, right? And in the, let me show you, so we have different kind of mechanism under passive. So this may be, so these are the aquaporins. So so this is our cell membrane. So this is the cell membrane. So this is cell membrane structure, and in that cell membrane structure we have aquaporins. So this aquaporins will allow some molecule to uh, get in and get out from the cell without the requirement of any energy. So that's why this aquaporin will facilitate the import and export, but without consuming, without utilizing any energy. So this is the part of passive transport mechanism, right? And another are, so just see in the figure. So if we have the gaseous molecule, and we can have the simple diffusion, and for this simple diffusion, what we must have, we have the higher concentration of the gas outside, and we have the lower concentration of gas inside the cell, then only this simple diffusion can be happened. So the one of the passive transport is by the simple diffusion. Then the second is facilitated diffusion. So if we do have the aquaporins, then this aquaporins can facilitate the diffusion. So this will come under facilitated diffusion. And third is osmosis. So if we do have higher concentration outside, lower concentration inside, then what will be happen? Then this higher concentration will move toward the low concentration, and this is how the molecule can be transport inside the cell. Okay, so we have simple diffusion, we have facilitated diffusion, we have the osmosis. So this will come under passive transport. And the another is active transport, where some energy will be required, right? So under this facilitated diffusion, we have this aquaporin. That aquaporin will facilitate this passive transport. So that's why the statement C, that aquaporins are specialized transmembrane protein that facilitate water transport, primarily function as water exporter, is the correct option. Okay, so next move to the next question. Okay, so we do have three classes of transport protein. So we talked about the aquaporins. Aquaporins are nothing but some transport proteins. But we do have three class of this transport protein: uniporters, and then we have symporters, and we have antiporters. So you have to tell me the difference between these three classes, how we can differentiate these three classes of transport protein. So I have four options. The first is uniporters transport one substance across the membrane, symporters transport two substances in opposite direction, and antiporters transport two substances in the same direction. The B option is all transport protein acquire ATP for function, with uniporter transporting one molecule at a time. Symporters and antiporters require energy input to move molecules. C option is uniporter transport a single substance, importer move two substance, and antiporter transport one substance while another move in the opposite direction. And D is uniporters, importers, and antiporters are passive transport protein that function only through facilitated diffusion. So how we can differentiate between these three classes of transport protein? So the answer is C. So what we have, if we have the uniporter, then we can transport one substance. If we have the same porter, we can move two substance in the same direction. And if we have the anti, then we can transport one substance, but the another will move in the opposite direction. Let me show you how. So, as I told you, we have the aquaporins, right? And these aquaporins are the part of the facilitated passive transport mechanism. So, because of this protein structure that we have in the cell membrane, that will help the molecule to get in or get out of the cell, right? And we have three type: uniporter, symporter, and antiporter. Okay, so here, if we have the uniport, if we have the uniport, then what we will have, we have the molecule, let's say glucose. And if we do have the higher concentration of glucose outside the cell membrane, then this glucose will be transport inside. If we do have the lower concentration of glucose, so this is uniporter. So in one direction, this is coming. Then antiport, if something is coming out, something has to go out. Okay, so this is what is known as the part of antiport. And symport, two molecules or some molecules can come inside, then this will be the part of the symport. So this is how we can classify three classes. If you have any doubt, then you can write in the chat box. I can repeat that. But the thing is, you must understand what we are talking about, what we are discussing about, because this is just for you people. Right? So these are just the explanation, and because I'm explaining, so I'm not reading the explanation. Okay, so the next is about the gas vesicle. So you have to tell me the incorrect statement, not the correct but incorrect statement about the gas vesicle. So we have four options. Gas vesicle provide buoyancy to cell, allowing them to position themselves optimally in a water column. Unlike typical biological membrane, gas vesicle membranes are composed of protein rather than lipid. Gas vesicles are impermeable to water and solute but allow gas to pass through. Gas vesicle can withstand high hydrostatic pressure due to their rigid protein structure. So what do you think? What is the incorrect statement for the gas vehicle? So first, let me tell you what do you mean by this gas vesicle. So we have, let's say, we have one cell. Okay, in that cell we have one organelle that is known as the vesicle, gas vesicle. So we have some organelles here, and that organelles are made up of the gas. So in which we have gas filled in these organelles. So what is the work of this gas vesicle is that whenever we have so let's say we have pond, right? So we have pond and we have sunlight. So for example, if we do have algae, then algae must have to be at the top of the water. Okay, so that photosynthesis can be happen. So what will happen, this gas vesicle will provide the buoyancy, so in such a way that this vesicle will allow this cell to float on the top, will not go to the bottom. So if this is the bottom, so what will happen, this gas vesicle will facilitate this cell to float or to be at the top of the river, so that it can photosynthesis can be happen, because cell needs less sunlight. If we do not have the gas vesicles, then the cell will sink at the bottom, and if this will be at the bottom, then photosynthetic activity will be reduced highly. Okay, so this is the work of the gas vesicle. If you give me B option, but the option is The option is D, because this was incorrect statement, not correct. So this was the right, that it will give you the buoyancy to cell. As I told you, the cell must be at the top of the river, so that sunlight can come and the photosynthesis can be happened, and this we can do using the gas vehicle. This gas vehicle will make the cell very lighter, so that it will provide buoyancy. So this is the right statement. Then gas vesicle are composed of protein rather than lipid. This is also right, and these are the right statement, not the incorrect. And these are impermeable to water. Why impermeable? Because this is providing you the buoyancy. If we do have water inside this gas vesicles, then this will be heavier in nature, so how it will provide the buoyancy to the cell? So it is impermeable to water, and that's why it just allow gas to pass through. And this is again the right statement, but this is not right statement. So that's why D is the right answer, because the question was what statement about the vesicle is incorrect. So let me show you here. So this is our cell, right? And in that cell we have this gas vesicle. We have the gas vesicle. So what will happen? And this is, let's say, a river body. Let's say this is the river body. Then this gas vesicle will allow this to come to at the top. So this will give you the buoyancy. This will compete against the gravity and the drag force. So that's why this cell will come up and will help for the photosynthesis. So this is primarily the work of this vesicle. Okay. So now coming to my next question. In the bacterial cell, so let's say this is the bacterial cell, they also have the endospores. And you have to tell why endospores are highly resistant to extreme environmental condition. So these are highly resistant to endospores, and these endospores are the part of the sexual reproduction in the bacteria. Most of the time bacteria have this binary fission, but in some cases the bacteria can undergo for this formation of the spores, and these spores are the endospores, and these are highly resistant to extreme environmental conditions. So whenever the condition will be favorable, these endospores will start to reproduce. Okay, until that this will be in inactive stage, right? So the question is about endospore. Then we have four options. Endospores contain large amount of water, preventing them from drying out. The presence of dipcholinergic acid, that is DPA, and high calcium ion concentration contribute to their stability and resistance. C is endospores have lipid rich exosporium that prevent the entry of harmful substance. And D is they are highly permeable to staining dyes, allowing easy visualization under light microscope. But the question is why these are highly resistant to extreme environmental condition. So first you have to see what do you mean by this endospores. So most of the time they have this DNA or the genetic material in the cytoplasm. So this is our bacteria, and in that cytoplasm we have the genetic material. Then this genetic material will copy. Then we do have the two genetic material, and then what will happen? This genetic material will have some covering, or this will have some protective layers. And then what will happen? This bacterial cell wall will be lysed or breaking down of this cell wall, and this is how this endospore will be released. So this is the endospore. Okay, and let me show you the next slide. So this is the structure of endospores. So what we have in the outer, we have the exosporium, we have outer coat, we have inner coat, we have cortex, we have DPA, we have ribosome in it, DNA, and we have some proteins, inner membrane and the core wall. So we have a lot of thing in endosporium, and this DPA helps this endospores to survive in the extreme condition until the condition is favorable and it will start to reproduce. Okay, so the answer is this one. So this resistance is highly due to DPA and calcium ion, which helps stabilize the spores DNA and protein against damage. Okay, so answer B is the right one. So you got to know that what do you mean by this endospore and why this endospore are like able to survive in the extreme condition. Okay, the next questions are about the technique by which we can visualize the component of the cell. So, for example, if we want to see the cell wall, if we want to see the mitochondria, if we want to see the genetic material of the cell, then we do have different techniques by which we can visualize this. Then we have some staining techniques. So, next coming 5 or 6 question will be on the staining technique only. So if you want to visualize the bacterial endospores, then what kind of staining we will use? Then we have 4 options: crystal violet, then malachite green, and then methylene blue and eosine. So what kind of staining we will use if we want to visualize this bacterial endospores? So we will use the malachite green, and let me show you how we can do this. So we have bacterias. We have a lot of bacterias. So we have the bacterias, and if we are using this stain malachite green, then what will happen? This will give the green color to the bacterias. Okay, this will give you the green color to the bacteria. And if you are steaming, if you are giving the steam for 5 minutes, and then you are washing this, then after washing, what you will get, the stain only will be retained under the endospores, but the everything outside of this bacteria will be washed off. So for example, you have this bacteria and you have this endospore. So this endospore will be stained only using this. Whenever you are washing off this slide, then this malachite green will give you the green color to this endospore, and this will be colorless. Okay, but whenever you are counter staining it with the safranin, then what will happen? The outer things of the bacteria will have the pink color, or let's say red color, but the endospore will have the green color. So this is how we can see. So here, if you will see, this is whole bacteria, and this is the remaining bacteria thing, and this is the endospore of the bacteria. So this is how you can see the endospores of the bacteria using malachite green. So malachite green is used in endosperm staining because it penetrates the tough spore core coat. We talked about the structure of endosperm spore also. So this malachite green can penetrate this spore coat and stain the endospores, which are then counter stained with safranin to distinguish vegetative cells. So this is how, if you want to see, if you want to visualize the endospores, then what you can do is first stain with the malachite green, then steam it, wash it, and then counter stain it with the saffronin. Then what you will get, the green color will show you the endospores, and the red color or pink color will give you the other part of the cell. So this is how you can identify, you can visualize the endospores. Okay, the next question is, which primary stain is used in the gram staining technique? And the options are India ink, methylene blue, crystal violet, and Sudan 3. So we have two type of bacteria: gram positive bacteria and gram negative bacteria. And if we want to distinguish, if we want to see, if we want to differentiate what kind of bacteria we have, then we have some staining technique based on which we can differentiate either it is gram positive or gram negative bacteria. So what we will use, we will use crystal violet stain. So let me show you how we can do this. So if we have some bacteria, if we have, let's say we have some bacteria gram positive and gram negative, but we do not know initially what kind of bacteria these are, whether these are positive or negative. So what we will do, first we will have a bacteria, then we will stain it with the crystal violet. Okay, so this will give you the violet color to gram positive and to the gram negative bacteria. Okay, then what we will do, we will have this iodine treatment. So what this iodine treatment will do, this will fix uh, and this will fix the bacteria on the slide. Okay, then what we will do, we will wash it, we will decolorize this, we will wash it with the alcohol. Then whenever we will wash, then what will happen, this will become colorless and this will have this violet color. Then if we are staining, we are doing counter staining with the saffronin, then this will have the pink color and this will have the violet color that has been retained of this crystal violet. So this is how, and if we have the pink color, then this will be the gram negative, and if we have the violet color, then this will be the gram positive bacteria. This is how we can differentiate. And how this is happening, that there is like, this is giving the colorless whenever we are washing, then this will be colorless and this will be with color. So this is because of the difference in the cell wall of the bacteria. We already have talked about the peptidoglycan, right? So in gram positive bacteria, we have thick layer of peptidoglycan, and this peptidoglycan will retain this crystal violet. But in the gram negative bacteria, we have very thin layer of the peptidoglycan in the cell wall, and based on that, because it is very thin, then all of this crystal violet stain will be removed after washing. But because we have very thick thick peptidoglycan layer in the cell wall of the gram positive, so violet will be retained. So that's why this is giving you a different color, and based on that you can identify whether your bacteria is gram positive or gram negative. Okay, so the main primarily stain is crystal violet, and the counter stain is saffronin. So the question giving you the answer is crystal violet. Okay, next is, if you want to see whether your bacteria is acid fast or acid slow, then again you will use different kind of dye or different staining method. So for example, if you have acid fast bacteria such as mycobacteria, so what kind of stain you will use? So we have four options: Congo red, carbol effusion, then C is bromophenol blue, and D is hematoxylin. hematoxylin. Yes. So what kind of stain we will use to detect whether this is acid fast bacteria or acid slow bacteria? So we will use the stain carbol-fission. Let me tell you this. So what will happen? So whenever you are using the dye, having this carbol fission, then this will give you the pink color. This will give you the pink color. Whenever you are applying this heat and acid or alcohol to wash, then decolorizer if we are using, then some of the bacteria will have will become transparent or will lose the color, and some bacteria will have this pink color. Then whenever we are counter staining it with the methylene blue, then some of the bacteria will have this blue color and some of the bacteria will have this pink color. So if we have the pink color, then this is acid fast bacteria, and if we do have the blue color, then these will be non-acid fast. Sorry, I was talking about the acid fast and acid slow, but no, was the acid fast and non-acid fast. So what will happen? This carbol fusion bind to the mycolic acid in the acid fast bacteria, and that will retain the stain even after decolorization with the acid alcohol. And this is how this will tell you whether your bacteria is acid fast or non acid fast, using the stain or using the dye carbol fusion. Okay. Okay, next question is, if you want to just see the negative staining to visualize the bacterial capsule, if you want to see the bacterial capsule only, for example, if you have a lot of things, a lot of like organic compound, and along with that organic compound you have the bacterial capsule. So if you just want to differentiate whether this is organic matter or you have the bacterial capsule, then what kind of stain you will use? Then we have four options: gram stain, India ink, methylene blue, and saffronin. So we will use India ink, and again let me explain you. So first we have the slide, right? Then what we will do, we will add India ink on it. Then we will prepare the smear. Then we will dry the smear, and if we will use the counter stain, that is crystal violet, and then what will happen, this will give you the capsules of the bacteria. Okay, so the primarily stain that we used is India ink. So India ink is used in negative staining where the background is stained but the capsule remain clear, making it easier to visualize the capsule structure. So that's how India ink is the right answer. Okay, next question is, again, if you want to visualize the nucleic acid in the bacterial cell, then how you will do that? What kind of stain or what kind of dye you will use? Then you have four options: Sudan black bee, nigrosin, then acridine orange, and carmine. So you can write in the chat box as well what kind of stain you will use whenever you want to visualize the nucleic acid only. So, for example, you want to extract out the nucleic acid from the bacterial cell, but first you have to see where is this nucleic acid. So, if you want to visualize that nucleic acid, then only you can perform the another activities. So, which dye or which stain you will use to visualize this nucleic acid? Any guess? So what we will do, we will have this acridine orange. Based on that, we can visualize the nucleic acid. So first you have to understand the unit of your nucleic acid. So the nucleic acid is made up of the nucleotides or polynucleotides. Okay, so you have this chain of the polynucleotide. This is the structure of your nucleic acid. And whenever you have the acridine orange, this acridine orange will form the complex with your polynucleotide. Okay, and whenever this will form the complex, this is giving you some color. Okay, and this is how you can differentiate between the cell and the nucleic acid inside the cell. So acridine orange bind to DNA and RNA. So nucleic acid is nothing but the genetic material. So this will bind to the DNA and RNA, and this will give you the light or fluorescence under UV light, because this is making you making the complex, making is useful for detecting nucleic acid in bacterial cell during fluorescence microscopy. Okay, so the basic principle is that we have acridine orange. We have, like in nucleic acid, we have the genetic material. This can be DNA, this can be RNA, and this DNA or RNA will make the complex with the acridine orange. And whenever we will see that complex under UV light, then this will give you the fluorescence phenomena, and this is how you can differentiate between genetic material or nucleic acid from cell. Okay, the next question is, okay, which of the method can simultaneously measure both cell size and cell number with high accuracy? So, for example, if we have some kind of bacterial infection, then you people know that we undergo through this tissue culture, right? So in that tissue culture, what we see, the type of bacteria, the we try to identify the bacteria which is giving you the infection, and even we are trying to see the characteristic of the bacteria, or the bacterial cell size, the bacteria number. So how many numbers we have of that particular bacteria. So we may adopt some method by which we can measure both cell size and cell number with high accuracy. So we have four options: hemocytometer, flow cytometer, then Petro Foster counting chamber, and then spread plate method. Someone gave me C answer. So let me show you. This is flow cytometry. Note the Petro Foster. Let me explain you how. So whenever we are talking about this hemocytometer and Petri Foster chamber, here we can count the cell. We can count the cell, but this will be manually. Okay, then this spread plate method also estimate this viable cell count. But if we are talking about the flow cytometry, this is very advanced technique where we can visualize both cell size and cell number using laser based detection. So this is not the manually. So the answer that you gave me, C, this is also the method, but also the method to measure cell size and cell membrane, but this is manual method. But the question is with high accuracy, and for the high accuracy we will use the flow cytometry. So your answer was right, but you just need to see whether like this is with the high accuracy or with the low accuracy. So if it is with the high accuracy, then we will go with flow cytometry. Okay. Okay, next, coming to the next question: which instrument is most suitable for counting bacterial cell but cannot determine their size? We talked about this flow cytometer. So this can both like count and even determine the size. So this is not the answer, but we have another three option. Coulter counter, hemocytometer, and the spectrophotometer. So we have the hemocytometer. So this hemocytometer is most suitable for counting bacterial cells, but it cannot detect the size. So here I can show you. So we have slide, right? We have a slide, and in that slide we have one cover slide. But here what we have in that slide as well, so there is some partition, so we have square within that. So within a single slide we have thousand of subdivision and thousand of square. And if we are taking one square, so let's say this is the one square, in that one square we can count how many cells we have, and based on that we can estimate like how many cells we may have in whole slide. Okay, and this is how we can calculate. So this is the manual counting device. Hemocytometer is the manual counting device, and for estimating bacterial and eukaryotic cell membrane, and it doesn't provide size measurement. Okay, but the coulter and the flow cytometer, these both can measure both size and number. And if we are talking about the spectrophotometer, then this can give you the cell density using optical density measurement. So we had four methods that we asked: coulter, hemocytometer, then flow cytometer, and then spectrophotometer, and the hemocytometer is the right answer, right? Okay, which technique is least effective for accurately determining bacterial cell concentration? So let me tell you, this is the optical density measurement, and because this provide an indirect estimation based on turbidity, but does not differentiate between live and dead cells. So if you people know the principle of spectrophotometer, a light will be given to that slide, and if like based on the absorbed light, this will tell you the density of the bacteria. But if it will not able to differentiate whether this is live bacteria or dead bacteria, because this is just based on the turbidity. This is not telling you whether your bacteria is live or dead. So that's why this is very like least effective for determining the bacterial cell concentration. But if we are talking about the another techniques, then they provide more direct and accurate bacterial cell count. So that's why C answer is the right one. Okay, now again we are coming back to the cell biology, and we are talking about different macromolecules, micromolecules, and how these macro and micromolecules uh, are making the whole cell, right? Then the question is, which macromolecule primarily makes up the structural framework and catalytic machinery of the microbial cell? So we have four options: carbohydrate, lipid, protein, nucleic acid. So what kind of macromolecule will make up the structural framework and catalytic machinery? So from catalytic machinery, it is very clear what kind of macromolecule we have for the catalytic machinery. So this is protein. So whenever you are talking about the catalyst or enzyme, most of the catalyst and enzymes are made up of the protein. So whenever you have any option like this, then you can blindly go to the protein. So for example, we have any type of enzyme, for example, the enzyme that we need for the replication, the enzyme that we need for the transcription, or any kind of enzyme that we have, or that catalyst we have, these catalysts mostly are made up of the protein. So that's why protein is the right answer. Here, if you will see, I gave you some, like, this is not direct description about the question, but something like you should know the percentage of element for the cell. So if we are talking about the carbons, then carbon is 50% in the cell. If we are talking about the nitrogen, then 14% of the total cell is made up of the nitrogen. Then we have oxygen, hydrogen, so we can give the proportion of the each of the element in the cell. And for the explanation, protein constitute up of the 55% of the dry weight, and these serve as the primarily structural component of the membrane and ribosome, and also act as enzyme that derive metabolic processes. So this is how the answer will be protein. Okay, the next question is, which of the following biomolecule is least involved in forming microbial cell structure but play a crucial role in energy storage? If you want to ask some question, because Ravi and Kongi raised the hand, if you have any question, you can write in the chat box. Anyway, so can you tell me the biomolecule that are least involved for the structure but play critical role in energy storage? So this is lipid, and let me show you. So we have four major or four micro biomolecule: lipid, nucleic acid, carbohydrate, and protein. Yes, sorry, this is lipid. And here, if we have the lipid, then lipid are like most of the time they are used for the storage. If we have the nucleic acid, this is for the storing the genetic information. All the genetic information are stored in the nucleic acid, or you can say DNA, RNA. If we are talking about the carbohydrate, then we have a lot of carbohydrate for the energy purpose. Then we have the protein. These protein are used for like this protein can act as the enzyme or catalyst. Okay, so this is how we have four major macromolecule, and each of these the work of each of these macro biomolecule is well defined. So here, if you will see, protein are made up of amino acid. From very first slide, I told you that we have the amino acid. This is the basic structure of amino acid. If we have the sequence of amino acid, then this will give you the protein. Okay. Then we have the structure of fatty acid. This, if we do have the uh, if we have the sequence of the fatty acid, this will give you the lipid. Then we have the sugar, we can have the glycerol and monosaccharide. And if we do have the nucleobases, and then this nucleobases can, this is the subunit for the nucleotide, and then the chain of the nucleotide will give you the DNA and RNA. And this lipid are made up of fatty acid and glycerol, and this will help you for the storage, right? And here also it is written lipid are primarily involved in energy storage, and in contrast proteins make up most cell structure, carbohydrates serve as cell wall component, nucleic acid store genetic material. So this is how we have four macro biomolecule and the function of these four. And the next question is, which macromolecule is essential for the protective coat of virus and many cellular function in microbes? So options are lipids, carbohydrates, proteins, and RNA. The protective coat of virus is made up of what? Lipid, carbohydrate, protein, RNA. So this is made up of protein, and let me show you how. So this is a basic structure of your virus, right? And viruses have the genetic material. This one is the genetic material, and this genetic material is covered with the protective layers. These layers are made up of protein. So it can be spike protein, it can be nucleocapsid protein, it can be envelope protein. So we have the genetic material in the form of nucleic acid inside the virus, but these are protected with different kind of protein, membrane glycoprotein. So the question, the answer will be viral capsid, or the viral covering is made up of protein which protect nucleic acid and facilitate host cell attachment. Or even if we are talking about the enzyme transporters, we talked about that aquaporins, right? So aquaporins are the transporters. So we have enzyme transporters, or even the structural components are made up of proteins. So this is how you can give the answer that viral capsid are made up of protein. So the answer protein is right one. Okay, so so let's say we have this bacterial cell, and we have some organelles into it. So let's say we have ribosome, we have the genetic material into it, we have some another organelles. So which of the following organelles has its own DNA, produce energy, and is believed to have originated from ancestor prokaryote? So let's say this is not the bacterial cell, if this is the animal cell. If this is the animal cell, so animal cell is the eukaryotic cell, right? So you have to tell me which of the organelle. So you have to tell me the name of the organelle, either this or this or this. So the organelle that we have inside the cell of, let's say, animal or plant cell, that have its own DNA, and that will produce the energy, and that is believed to be originated from the prokaryotes. So you have to tell me what kind of organelle we are talking about inside either the animal cell or the plant cell that have its own DNA. So the options are lysosome, endoplasmic reticulum, mitochondria, and peroxisomes. You know that within the cell of, let's say, animal cell or the plant cell, we have the genetic material in the nucleus. Okay, but we have some another organelles as well. In that organelles, they also have the DNA into that organelle. So you have to tell me the organelles out of these four options, which organelles have its own DNA, not just in the nucleus. So the answer is mitochondria. And let me explain you, see the last line. It is believed to have originated from the ancestral prokaryotes. So let me show you how. So we have the cell. This cell can be animal cell or plant cell, and this is the nucleus. In that nucleus we have the genetic material. Another organelles we have, so we have cytoplasm, we have mitochondria, and we have Golgi apparatus. We have a lot of organelles into that cell. But if we are talking about this mitochondria, this mitochondria have its own DNA. Mitochondria have its own DNA. So, and the question also had like this na, ki, it is originated from the prokaryotes. So there is one theory, that is endosymbiotic theory, that claims that this mitochondria is nothing but one the prokaryotic cell, and this prokaryotic cell entered into this cell, and this is how this prokaryotic cell changed into mitochondria, and that's why this mitochondria have its own DNA. So it was nothing but one prokaryotic cell initially it was, and that gave rise to mitochondria, and that's why this mitochondria have its own DNA. So this comes under endosymbiotic theory. Let me show you a little more detail about this. So let's say initially we have the prokaryote, and aerobic bacteria entered into this prokaryote, and this is how this gave rise to the mitochondria. Okay, and because this aerobic bacteria already had the DNA, then this prokaryotic cell will have genetic material different from the genetic material that has been uh, entered using this aerobic bacteria. So that's why this cell have two genetic material. One is this one, and the one is inside this mitochondria. Then another cyanobacteria entered into this, and this cyanobacteria gave rise to chloroplast. So it is said that initially we had the prokaryote, then some cells were eaten up by the prokaryotes, and that's why this cell already had the DNA material. So we have some organelles, for example mitochondria, for example chloroplast, that have its own DNA, because these were initially the bacteria that has been eaten up by the another prokaryotes. So that's why these organelles have the DNA uh, uh, along with the nucleus, and that nucleus also have the genetic material. So this is nothing but the endosymbiosis hypothesis. So prokaryote ingested some aerobic bacteria, and this aerobic bacteria were protected and produce energy for the prokaryotes, and this is how we have some organelles inside the animal cell or plant cell that have its own DNA. Okay, the next question is, which of the following characteristics is shared by the mitochondria and cytoplasm but not by the nucleus? Options are possession of double membrane, presence of DNA, ability to divide independently, regulation of gene expression. So as I told you, we have three type of genetic material that we have inside our cell. One is the DNA material that we have inside the mitochondria. One DNA material that we have inside the chloroplast, and the another genetic material that we have inside the nucleus. Okay, so we have three genetic material that we have inside our cell, in the mitochondria, in the cytoplasm, in the nucleus. Now, the question is, which of the following characteristics is shared by the mitochondria and chloroplast but not by the nucleus? And the answer is ability to divide independently, because this mitochondria and cytoplasm were nothing but were the prokaryotic cell, right? And this prokaryotic cell can divide independently, but not by the nucleus. Nucleus have to undergo under this mitosis and meiosis, and based on that only it can be divided. So mitochondria and cytoplasm has the ability to divide independently. So unlike the nucleus, it relies on the cell cycle for division. Mitochondria and chloroplast replicate independently, similar to the binary fission, but here the DNA have to undergo under division, that is mitosis and meiosis. Okay, which feature of mitochondria provides the strongest evidence for their bacterial origin? We talked about the endosymbiotic theory, in which it was said that initially we had the prokaryotic cell. One prokaryotic cell has been eaten up by this cell, and that's why this cell is now the mitochondria, or let's say chloroplast. So what feature of the mitochondria provide the strongest evidence that it has been originated from the bacterial? So we have four options. They generate ATP, They are present in all eukaryotic cell. They contain 70s ribosomes similar to prokaryotes. They are surrounding by a double membrane. What do you think? What is the right answer for this? So the right answer is they contain 90s ribosome. So let me show you. So we have this in the left hand side. What we have, this is the structure of the mitochondria, and mitochondria have the matrix. In that matrix it has its own DNA and it has its own ribosome, and this ribosome is different from that ribosome that we have in the eukaryotic cell, in the cytoplasm of the eukaryotic cell. And if we are talking about the prokaryotic ribosome, based on the size, based on the shape, it has been known as 70s. Okay, and the eukaryotic ribosome is 80s. So in the mitochondria we have 70s ribosome, but in the cytoplasm we have 80s ribosome. So what we can, and bacteria cell also have 70s ribosome. So what we are estimating, that this mitochondria was nothing but the bacterial cell, because this mitochondria have the 70s, and prokaryotic cell also have this 70s ribosome. Okay, so that's why this is giving you the evidence that this mitochondria is nothing but it was initially the bacterial cell or one cell that has been eaten up by a different cell, right? Okay, the next question is based on the microscopy. As I told you, we will talk about the cell biology, and then to visualize different organelles, to visualize the cell, we need to have some instrument. Okay, and that instrument have some characteristics that have the magnification, that have some spatial resolution. So the question is, calculate the wavelength of light if the diameter of the smallest resolvable object size is 312 nanometer and the specimen is observed with the 60x oil immersion lens having the numerical aperture of 1.40. So can you calculate the wavelength of the light that can resolve, the object having the size of 312 nanometer? So how we can do this? We have a very easy formula, and that formula is d. This is the distance between 2 points, and this distance is equal to 0.5 multiplied by the wavelength divided by the numerical aperture. So what we have, we do have the distance, and that distance is 312 nanometer, and we also have the numerical aperture, that is 1.40, and that's how we can calculate the wavelength. And this wavelength will be nothing but just put the value here, and then you will get the wavelength, and that wavelength will be 780 nanometer. So this is how you can estimate the wavelength. Okay, so different microscope we need for the different purpose. So if you want to observe the internal structure of the virus, and that have the size 80 nanometer, so what kind of microscope you need? So we have option phase contrast microscope, we have option dark field microscope, uh, transmission electron microscope, and fluorescence microscope. What kind of microscope we need if we want to observe the internal structure of the virus? So to see the internal structure of the virus, the microscope will be transmission electron microscope. So you have to remember it. each of these microscope have some advantage and some disadvantage, and based on our purpose we can use different kind of microscope. So if you want to see the internal structure of virus, you will use the transmission electron microscope. So here if you will see that such kind of structure we can see using transmission electron microscope. So the viruses are typical in the range of 20 to 300 nanometer, and this is below the resolving power of the optical microscope. Optical microscope have resolving power of just near to 200 nanometer, and below that we need higher resolution microscope, and this will be transmission electron microscope. So that will use much shorter wavelength than visible light, and that will allow to resolve structure at the nanometer scale. So this is how transmission electron microscope will be used if we want to see the internal structure of the virus. This is again the question on gram staining procedure, and you have to tell me what color we will have after applying some dye or some chemical. So we have alcohol acetone, we have crystal violet, and we have safranin, and you have to tell if our bacteria is gram positive, then after applying alcohol what kind of color it will show. If we are applying the crystal violet, then what kind of color it will give, and after safranin what kind of color it will give you if it is the gram positive bacteria. So, what was the principle of the gram-positive bacteria staining? Is that gram-positive bacteria have the thick layer of peptidoglycan. So, first, what we do, we stain it using the crystal violet, and this crystal violet give that the violet color. Then, what we will do, we will wash it by the alcohol, but because it had the thick layer, then this will give you the same color. And then if we are counter staining it with the safranin, then also it will give you the same color. So that's how D answer is the right one. And let me show you here. If you have the gram positive bacteria, then we have some bacteria, and if you are applying this crystal violet, this is giving you purple color. Then you have iodine and the ethylene alcohol. This will if it will be the gram negative bacteria, then this will be become colorless, but if it is the gram positive bacteria, this will retain the color. If we are counter staining it with the safranin, then this colorless or transparent bacteria will take the color pink, and the violet color will be as it is for the gram positive. So this is how the sequence will be this one only. So this crystal violet will stain the cell purple. Alcohol acetone does not remove. Is it that my slide is not visible? Let me see. I think my slide is visible, isn't it? Anyone else? My slide is visible to you? Okay, this is visible. Yanendra, it may be that you may have some network problem, but the slides are visible. So this is how we can stain and we can differentiate between gram negative and gram positive bacteria. This is again the same question. Which of the following describe the effect of over decolorization on the gram positive bacterial sample? So what will happen if we are applying this over decolorization, or if we are washing it with the excessive alcohol acetone? Then what will happen? Cell appear purple after counter staining. Cell appear pink due to loss of crystal violet stain. Cell appear colorless after crystal violet staining. Cell appear green due to iodine interaction. We are talking about the gram positive bacteria sample. So this will appear pink due to the loss of crystal violet stain, because if we are doing the over decolorization, if the colorization is in optimum condition, then this will give you the purple color. But if this is very excessive in nature, then this will give you the pink. Let me uh, which staining step is crucial for distinguishing gram positive and gram negative bacteria? And this will be the B, that is alcohol acetone decolorization, because this, whenever we are washing our cell after staining, then the gram negative bacteria will give the colorless, and the gram positive bacteria will have the purple color. So this is very important step. Uh, to make the difference between gram positive and gram negative. I'm not going here. Okay, which of the following pair is mismatched? So we have different type of microscopy and the use of the microscopy or the principle of microscopy. So you have to tell me which of the pair is mismatched. So we have electron microscopy, phase contrast microscopy, bright field microscopy, and fluorescence microscopy. So you have to see the principle or the use of this microscopy and which one is giving you the incorrect pair. So fluorescence microscopy is giving you the incorrect or mismatch. Uh, just see, if we are using the electron microscopy, then we use electromagnetic lens. If we have the phase contrast microscopy, then the principle is we have different refractive index, and exploiting that different refractive index, uh, we can have this phase contrast microscopy. Then in the bright field microscopy, we specimen appear dark against a light background. And for the fluorescence microscopy, this is wrong. But why? Because in fluorescence microscopy, we typically use UV light, not the visible light, for to excite fluorescent dye, and that's how the specimen glow under the microscope. So we are not using the visible light for the excitation, but we are using the UV light to excite, and that's how D option is incorrect, or you can say a mismatch. Okay, next question is also about the microscopy, and you have to tell me which one is the incorrect. So atomic force microscopy provide 3D surface image at the nanometer scale. SCM, that is the scanning electron microscopy, provide internal detail of the specimen. Phase contrast microscopy allow viewing of live unstained cell. Dark field microscopy is useful for visualizing thin bacteria such as Treponema pallidum. So what do you think? What is the right answer for this? So the incorrect statement is about the scanning electron microscopy. As in the last question, we discussed that this is the transmission electron microscope that is giving you the internal detail of the specimen, but this scanning electron microscope is giving you the just the surface detail of the specimen, right? So here also I have the explanation. This scanning electron microscope provides surface detail, not internal structure. But the transmission electron microscopy is used for the internal cellular detail. So that's why this is incorrect statement. Next question is, which of the following statement is incorrect regarding microscope component? So you can read this And let me tell you the answer, that objective lens directly capture light from the illuminator is the incorrect statement. Why this is incorrect statement? Because the objective lens does not capture light directly from the illuminator. It collect light that has already passed through the specimen. So this is how this is the incorrect statement. Okay, this is something logical question. Which of the following is not a primary purpose of fixing bacterial cell to the slide? So as I told you, uh, we have the cells on the slide, then we will stain it, we will wash it, we will counter stain it, and this is how we can differentiate, or we can see the different component of the cell. But the question here is, whenever we are fixing the bacterial cell to the slide, then what is not the primarily purpose of this fixing? Then we have four options: preventing the cell from being washed off during staining, enhancing contrast for phase contrast microscopy, killing the bacteria to prevent contamination, preserving cell morphology for microscopic analysis. So what is not the primary purpose of fixing bacterial cells to a slide? So the answer is enhancing contrast for phase contrast microscopy. We fix cells so that we can avoid that cell will be washed off. So, and we also prevent the contamination. So for example, if we do have a very infectious bacteria, then we will try to fix that bacterial cell on the slide so that it is not spread in the laboratory. Then we are trying to preserve cell morphology so that we can go for the microscopic analysis. So these are the primarily purpose, but not the enhancing contrast for phase contrast microscopy. um Let me go to the next. Okay, which is one potential drawback of heat fixing bacterial cell to a slide? So what is the a disadvantage for this heat fixing bacterial cell to the slide? Options we have is, bacterial cells may become distorted, bacterial cells remain alive and can still glow on the slide, heat fixation increase the motility of the bacterial cell, heat fixation remove the cell wall of the bacteria, gram-positive bacteria. So what is the disadvantage of fixing the bacterial cell on the slide? Heat fixing. So we are giving the heat so that bacterial cell can be fixed on the slide. So what is the disadvantage of this technique? The disadvantage is that bacterial cell may become distorted due to heat fixation, right? So, heat fixation can cause distortion in the bacterial cell shape due to excessive dryness and heating. So, this is the disadvantage of heat fixing the bacterial cell on the slide. Which of the following statement is false regarding chemical and heat fixation? Chemical fixation is better at preserving fine cellular detail than heat fixation. Heat fixation is commonly used for bacterial smears before gram staining. Chemical fixation always preserves cell viability. And D is, heat fixation is simpler and faster method than chemical fixation. And what you need to tell is the false statement regarding chemical and heat fixation. So the answer is chemical fixation always preserve cell viability. No, this is not correct. This can kill the cell, and that's why this chemical fixation is not giving you the detail of the live bacterial cell. So this is the incorrect statement. So chemical fixation also kill the cell, though it may maintain their structure better than heat fixation, but it will kill the cell as well. Okay, so this is the incorrect statement. I think we do have a lot of question on this gram stirring technique. So what I think I should skip some question. Mhm. This is also based on this. This is based on, again, you can see the slides actually at last, because I'm just skipping right now the question on the staining. Okay, which of the following directly affects the resolution of a microscope? So we have four options: the wavelength of light used for illumination, the total magnification of the microscope, the size of the earpiece lens, and the thickness of the specimen. So if you want to increase or decrease the resolution of the microscope, what component or what variable have the direct impact on the resolution of the microscope? So the wavelength of the light that we are used for the illumination is giving you the direct impact or direct effect that will affect the resolution of the microscope. So if you see here, resolution depend on the wavelength of the light used. So if we have the shorter wavelength will provide you the higher resolution comparison to the longer wavelength. And magnification alone does not improve resolution. No, this is not the correct statement. And another are also like will not directly impact but indirectly. Okay, what happens when the numerical aperture of the microscope lens increases? So the resolution improve, allowing finer detail to be distinguished. The total magnification decreases, the wavelength of light used for illumination increases, the working distance between the lens and the specimen increases. So what will happen whenever like the numerical aperture of the lens increases? We had one formula, we had one numerical question, right? Based on the numerical aperture and the wavelength and the diameter. So what will happen? The resolution will improve and this will allow finer detail to be distinguished. So this will be the correct answer. Okay, which of the following is beyond the resolving power of a standard optical microscope? The bacterial cell that is 1 micron in size, 2 points separated by zero point 0.3 microns, 2 points separated by zero point 0.1 microns, yeast cell that is 5 micron in size. So we have optical microscope, but optical microscope have some limitation. So and that has some limitation to resolve the distance. So at what distance the capacity or the ability of the optical microscope is limited? So the answer is 2 points separated by zero point 0.1 micron. So if we want to resolve the distance or if we want to resolve the distance between 2 points and that 2 point have the distance of zero point 0.1 micron, then this is beyond the capacity of the optical microscope. We need advanced microscope that can resolve the distance between these 2 point, and this 2 point have the distance of zero point 0.1 micron. So up to only my zero point 0.1 micron distance between 2 point, optical microscope can help us, but beyond that, if the distance between these 2 point is lesser, then we need advanced optical microscope. Okay, which of the following macromolecule is the most structurally and functionally diverse in a cell? Carbohydrate, protein, lipid, nucleic acid. So this is protein. Protein is like, as I told you, protein is made up of amino acid, and we have 20 amino acid, and this will give you the uh, a chain of the amino acid using the polypeptide bond, and this is how the protein is formed. And this is giving you just the primary structure of the protein. Then this primary structure will have some bonding that will give you the secondary structure. Then we also have some different kind of bond that will give us the tertiary structure. Then this tertiary structure can also be modified into the quaternary structure. So what we are getting, that we have 20 amino acid. 20 amino acid can give you the diverse form of the protein. The first thing. The second thing is different bond within this amino acids can give you a very diverse structure of the proteins. This can give you the primary, secondary, tertiary, and the quaternary protein structure. So, and the function is very different. Like protein can help us for the catalytic activity, for the enzyme activity, for the transporters. So it is very diverse structurally and functionally. So this is very important macromolecule that we have in the cell, and that is protein. Okay, which of the following macromolecule makes up the largest proportion of the dry cell of the bacterial cell? Dry weight of the bacterial cell. Lipid, protein, RNA, carbohydrate. So this is protein. Protein make up approximately 55% of the dry weight of the bacterial cell. Lipid is 9 to 10, RNA is 20, and carbohydrate is 5. So this is protein. Which statement about protein in the cell is false? Proteins are polymer of amino acid. Protein play roles in enzymatic activity, transport, and structural support. Proteins are only involved in energy storage. Protein contributes significantly to the dry weight of the bacterial cell. From very first question, we talked about that protein are made up of the amino acid. So this is right answer. Then yes, Garindra, C is the right answer. Then we talked about that protein have the role in the transportation as well. Aquaporins are made up of the proteins. Then we also talked about that proteins are having the more weightage in the bacterial cell, because it is having the 55 proportion of the total dry weight. Then D is also not the answer. Then C is the right answer. Okay. Which of the following molecule contain both ribose and adenine? DNA and RNA, ATP and NAD, protein and phospholipid, glucose and fatty acid. That have the both ribose and adenine. So ribose is the sugar, adenine is the amino acid. So which molecule have these both things, ribose and adenine? So answer A and B and C and D. So if you will see, B is the right answer. Let me tell you why. If we are talking about the DNA, then in the DNA we have the deoxyribose sugar. We have the adenine, but we have the deoxyribose sugar. So DNA is not the answer. Then ATP, adenosine triphosphate. So this have the adenosine. So this have the adenine, and NAD also have the ribose sugar. So this is the right answer. Protein doesn't have the ribose. Protein just have the amino acids. Okay, so protein have the adenine. Adenine is the amino acid. So this is again not the right answer, because protein does not have the ribose. And then glucose and fatty acid doesn't have either this ribose or adenine. Adenine is the amino acid. Amino acid may be present in the protein and may be present in the DNA, RNA, and ATP. Okay. Uh, Dilip Kumar, just give me 2 minutes. I'm just finishing this. So that's why answer B is the right one. Then we have which of the following statement about nucleotide is false? Nucleotide is nothing. Nucleotide is the backbone of your DNA. So nucleotide have sugar. Nucleotide have phosphorus. Nucleotide have some amino group. So nucleotide have three things: amino group, or amino acid, then phosphate group, and the sugar. So you have to tell me like which statement is false. Then DNA nucleotide contain ribose sugar, as I told you. DNA means uh, deoxyribose sugar. So we have the deoxyribose sugar that we have. So this is the wrong statement. We have the deoxyribose sugar, not the ribose. Ribose sugar is present in the RNA. Adenine can be found in all of the following molecule except what? So we have amino acid adenine that we have present in the DNA, RNA, and ATP, but not in the glucose. So that's why C is the right answer, because this is not the molecule where adenine is present. Then you just need to tell which statement is the right. Then polysaccharide is found in the storage granules. Lipid are extensively found in the cell wall. And if you will see here, the first statement is true, but the second is absent, because lipids are also present in the cell wall. Okay. Then phospholipids are the major component of the cytoplasmic membrane. Ribosomes are only found in nucleus. The second statement is not correct, because ribosomes are not only present in nucleus but in the cytoplasm and in the mitochondria and in the chloroplast as well. Then bacterial cell is enclosed within a nucleus. No, we do not have nucleus within the bacterial cell. This is nucleic acid. Then this is the false statement. Peptidoglycan is the major component of the bacterial cell wall. This is correct. Then C is the right answer. Which of the following is not a function of nucleotide? Then this is actually nucleotide function is to store genetic material. So this question is little wrong. So this is the main function of the nucleotide. Nucleotide function is to store the genetic information. The following statement is false. Then which is that false? Nucleotide directly catalyzed biochemical reaction. No, protein catalyzed that biochemical reaction, not the nucleotide. So that's why this is the false statement. And what is not the role of nucleotide? Then nucleotide act as structural component. No, this is not the structural component. This is the main portion for the nucleic acid or the genetic material. Second, abundant macromolecule in the cell after water is protein. We already have talked about this. Then the fourth statement about the plasma membrane, then this prevent all molecules from entering or leaving the cell, because this is not the correct, because this is semi-permeable. Some of the compound can be entered, some of the compound can be exit, but not completely. So that's why this is false statement. This is again the same. Let me skip this. We already have talked about the active transport and the passive transport. So I'm just skipping this. True about the active transport, because active transport require energy, and that energy can be in the form of ATP. So it require both ATP, and that's how I finished it. Sorry for being late, but thank you so much for showing the patience. Thank you so much, everyone, and we will meet again from 6 to 8 next Monday. So thank you so much for showing me the patience. Thank you, Anu. Thank you, Alekhya. Thank you, everyone. Thank you, Radhika. Thank you, Ganesh. Thank you, Ankita. I can't read names so fastly, but yes. Shiva Mani, Ganesh Kumar, Kiran, Ganendra, Darshi, everyone, thank you so much. And we will meet again from 6 to 8 Monday. And I think we have 4 or 5 more classes, then we will finish this microbiology course as well. So right now, this is the dinner time. You people can leave and have a good dinner food. So thank you so much.